My name's Dave Gorkin. I'm an AP Giannini postdoctoral fellow at the University of California in San Diego. Before I was a postdoctoral fellow and before I even went to graduate school to pursue my doctorate, I was a high school teacher in New York City. It may seem that teaching high school and doing biomedical research are very different, but in fact it's the same thing that drives me to do both, and that is a very, very deep love of learning. And so in both jobs, in teaching and in doing science, I get to learn every day, and I get to be a part of others learning every day. And that passion for learning really shapes who I am as a scientist, because it means that curiosity is my compass. So I want to find an area where there are lots of open questions, where we don't really understand what's going on, and that's the direction I want to head in scientifically. And so in my postdoctoral research, the direction that my curiosity has taken me is to the inside of a cell's nucleus. And more specifically, I'm curious about how such a massive amount of DNA as the human genome can be folded up inside such a tiny space as the nucleus of a cell. So if you were to take the DNA from just one human cell and stretch it out end to end, it would be about 2 meters long, so in fact longer than my outstretched arms. So to give you an idea of scale, that would be roughly like taking a string 30 miles long, so extending from here at Stanford to the end of the pier at Santa Cruz, and then folding up that 30-mile string inside of a basketball. And the point I want to make is that the DNA is not just folded up inside of the nucleus haphazardly. It's very highly organized because it has to function inside of the nucleus. DNA has to replicate during every cell division. DNA has to get repaired when it gets damaged. And importantly, genes have to be turned on and turned off in response to the right cues and at the right time. And so this field of DNA folding is really undergoing a revolution, due in large part to technological advances. And we're now learning that processes like gene regulation, that is genes getting turned on and off at the right time, rely on very precise folding of DNA. Our DNA is organized into chromosomes and then into structures that we call topologically associating domains, or TADs for short, and then into structures like loops. And this is all necessary so that an individual gene can function in a very complex environment. We're also learning that if DNA folding doesn't occur properly, it can have very serious consequences for human health. So in just the past few years, we've learned that abnormal DNA folding can contribute to cancer. It can cause a variety of birth defects. And so far, these are only rare cases, but I think they represent the tip of an iceberg. And what I think these cases are signaling to us is that DNA folding may play a much wider role in human disease than we currently appreciate. So in my postdoctoral research, I've set out to answer a relatively straightforward but as yet unknown question about DNA folding. And that is, does DNA folding differ between individuals in the general population? So in other words, if you could look inside of my cells and look inside of your cells, would our DNA be folded the same? Would it be folded differently? And if it's folded differently, might that have something to do with our relative health and disease risk? And so to answer that question, we need a way to map DNA folding inside of cells. We need to know how the DNA is folding. And the traditional way to understand how things are organized inside of cells is to take cells and just look down a microscope at them. But unfortunately, our DNA is too small and its folding is way too intricate to view under a microscope. So as I mentioned earlier, we have a new set of technologies that have really revolutionized this field. And those technologies don't use a microscope. Instead, they use high-throughput DNA sequencing. And so these technologies, which altogether we call chromatin conformation capture, generally work like this. You start with live cells, and then we use molecular techniques that allow us to take pieces of DNA that are near each other inside the nucleus and actually stitch them together into a single molecule. And once we've stitched them together into that single molecule, we can then use DNA sequencing to measure how often any two pieces of DNA are near each other in the nucleus. And finally, we can use computational tools to reconstruct the folding of the entire genome in the nucleus. And so I've now used these techniques, this chromatin conformation capture, to map DNA folding in blood cells from 20 different individuals. And this study's still ongoing, but I have some important preliminary findings that I'd like to share with you today. The first is that we do indeed find thousands of regions that are folded differently between different individuals. And the second is that in at least some of these cases, it seems that these differences in DNA folding are caused by differences in DNA sequence between these individuals. And that's really important because if you take any two individuals who are unrelated in the population and you compare their DNA sequence, there are going to be millions of positions at which their DNA sequence is different. And what our data suggests is that those differences in sequence actually can change the way the DNA is folded inside of the cells. Now, we don't yet know whether those changes in folding might impact human disease or health, but we are currently investigating that. And so my larger vision, which I hope I've communicated to you today, is that by learning about DNA folding, we can have a better understanding of the fundamental mechanisms of disease. And ultimately, I believe that will help our ability to both diagnose and treat patients. And in the meantime, one thing I can tell you for sure is that even in such a tiny corner of nature as the inside of a cell's nucleus, there is plenty of complexity and intricacy and beauty to keep me and many others learning for a lifetime.